We'll now start with the Genentech team 1. We have Susan from Kenya. She's a lecturer of clinical pharmacy and pharmacology in BC University. She's also pursuing her PhD in pediatric oncology in Vrije University in Netherlands. We also have Sadik. He earned his PhD in pharmaceutical sciences in the University of South Australia, and he now holds a postdoc position in clinical health sciences. So this project is quite unique because it combines the concepts of both PKPD and PBPK. This project, we were provided with a molecule from the Genentech company, which happens to be a transporter inhibitor. And the team modeled the drug to quantify the in vivo magnitude of inhibition and to predict the DDI. We also like to thank Kenta so much for coming today. I know he's on vacation and he made time just to attend today and support our fellows. Thank you very much, Kenta. And the floor is yours, Susan and Sadik. Go ahead.
Thank you, Amira, for the nice introduction, and good afternoon, everyone. Welcome back from the tea break. So our project is on model-based assessment of the kinetics of porphyrin, which is CP1, and the genus biomarker of APT2024 in dose escalation study of weak APT1 inhibitor for prediction of drug-drug interaction. So myself and Susan, my partner, our mentors are Kenta and Sravanti from Genentech, and from APT Amira. Just a little bit of introduction. OATP1B transporters, actually, they are highly expressed in sinusoidal membrane of the human hepatocyte, and they mediate the hepatic uptake and facilitate elimination of their substrates. So in the presence of inhibition by different drugs, this can actually lead to significant clinical drug-drug interaction. This drug-drug interaction is actually significant, but there is no a good way of predicting. There is a challenge in predicting the clinical drug-drug interaction. What happens is, in the presence of the inhibitors here, the drugs cannot be easily uptaken by the hepatocyte, or it's not easily eliminated, so the systemic concentration of the drug will increase, leading to adverse effects. So because of these regulatory guidelines, including FDA and the European Medicine Agency and others also, they require, they recommend to do in vitro inhibition study for the transporters, so that to determine if the drug has risk of drug-drug interaction. But the methods that we have so far, they have some limitation, including the in vitro method and PBPK method. Because of this, there is an increased interest in using endogenous biomarkers such as CP1 for a prediction of drug-drug interactions. This is generally the decision tree, how we go with drug-drug interaction evaluation. The first one is for a new molecular entity. As I said earlier, the regulatory authorities recommend to do inhibition tests for the transporters. And then this static analysis, the R value is used, and if it is less than 1.1, there is less risk of drug-drug interaction, so no problem at all. But the question is, if it is greater than 1.1, this can indicate the risk of drug-drug interaction. But this static by itself is not adequate enough to capture the potential clinical drug-drug interaction. That means there are some false positive results associated with this one. So we need a better or a method that can really predict the drug-drug interaction. And that is the point where our project comes in, to conduct modeling and simulation using endogenous biomarker, and then to predict drug-drug interaction. And then based on the result, we can conduct dedicated clinical drug-drug interaction, or a label can be based on the PBPK readouts. So with the CP1, previous studies have confirmed that CP1 and probe substances, for example, such as pravastatin and rosuvastatin, and other probe drug substances have shown that similar pattern with CP1, confirming CP1 can be used as can be integrated into a modeling approach to predict drug-drug interaction, and that's actually what we did for our compound, which is anonymous internal Genentech internal molecule called GDCX, which is inhibitor of the OATP1B. These are the objectives, the aims and objectives. The first objective is to determine the magnitude of OATP1B in vivo inhibition based on the CP1 PK in the presence of the inhibitor, which is DDCX in this case. Secondly, to compare the estimated in vivo inhibition and in vitro inhibition of OATP1 by the GDCX. And finally, to predict the magnitude of transporter-mediated clinical interaction between the inhibitor and the probe drug rosuvastatin. So this is the method. The method is basically divided into two major parts. The first one is the estimation, and second one is prediction. We had the data from Genentech, which is concentration time profile of the endogenous biomarker CP1 in the presence of the inhibitor, along with the PK parameters. And then in SimSip, the PBPK, the concentration, the portal vein concentration of GDCX was predicted. And then this concentration was used in PK-PD model to estimate in vivo inhibition constant of GDCX against the transporter. So this part, the first part is PK-PD, and second one is the prediction part where we actually use the inhibition constant obtained from PK-PD was used and integrated into the PBPK model in SimSip, and finally the clinical drug-drug interaction between the inhibitor and the rosuvastatin was predicted. A little bit of information regarding the study design. This is first in human study, phase 1, a double-blind MAD multiple ascending dose study with 4 different groups or cohorts having different doses, 200, 400, 800, and 1,200, once daily dosing, 3 to 4 patients per group. And the PK sampling was done for the inhibitor as well as for CP1 at day 1 and then at day 50. That is the time at which the inhibitor expected to reach steady state concentration. This plot shows the CP1 concentration profile of individual patient in the presence of the inhibitor at different dose groups. And this one shows the mean CP1 concentration, as you can see, for different dose groups with standard deviation. And they show that there is increase in mean CP1 concentration across dose levels. So as dose increases, the level of CP1 in the plasma has increased. And this one is Emacs model, where it shows exposure and response, whereby the response is CP1 concentration, and then the exposure is the Cmax and AUC of the inhibitor. So as you can see here, there is an increase as the exposure increase, like Cmax or AUC. The maximum CP1 concentration has shown an increase with the Cmax and AUC. Saying this, I'll pass it to my partner, Susan, to discuss the other parts of the project. Thank you.
Thank you so much, Sadik, for the first part of the project. So we're going to go into the PKPD modeling, and this was our model structure, that we had a central department. We did only a one model, and as you can see, that the clearance, the non-hepatic clearance for drug was 12.9%, and for the hepatic clearance was only 87.1%. So literally what we're trying to see in this diagram is that most of our the hepatic clearance was higher than non-hepatic clearance. When you look at the parameter estimates, we were able to get the Ki from our PK model. The Ki was 6.95 from the right slide, the right diagram. This is how our normal model looked like. As you can see, you can be able to see our input, you can be able to see the compartments in that it was only one. And on your right, you can also still see the Ki as being 6.95. So this is what we got from our PK-PD model, but from the VPC, we could be able to see that the model described our data very well. And on the right, in the individual prediction and the C1 concentration of the observed, we were able to see that they correlated very well. As you can be able to see that the individual predictions and the observed data are all distributed along the same curve. So when we got our in vivo Ki and we also got our in vitro Ki, we were able to check the fold difference. And we realized that the fold difference was 5-fold. And this was in line with what was in literature. In literature, the fold difference ranged from 2 to 37. In our case, it was 5-fold, and so it was in line with what was in literature. So this difference between the in vitro and in vivo, with the in vitro being higher than the in vivo, is not well understood in a way, but as from literature we've learned that there's some postulations about it. But the difference itself just shows why we need to be doing this CP1 studies, to be able to be getting CP1 data and being able to actually check what the Ki is. Afterwards, then we go to the PBPK model now. We already got our Ki, in vivo Ki, and now we put it into the PBPK model of superstatin and GDCX and SimSip. And this is what we got as the output. So what you see on this number one is the summary page. Basically, the whole of this slide is the summary page or snippets of the summary page. And you can be able to see that we had a workspace. We had the substrate here, which was superstatin, and the endogenous biomarker, which is CP1. And from there, you can be able to see that we had different physicochemical properties of the substrate, the inhibitor, and the endogenous biomarker. And you can be able to see anonymize the inhibitor because it's still an internal molecule. But then you can be able to see that we had to put in the physicochemical properties. And then afterwards, you can be able to see the model structure here. We used, of course, the Adam model for both the substrate and the inhibitor. And we were able to, if you look at the distribution model, you can see that for the substrate, it was a full PBPK model. For the inhibitor, it was a minimal PBPK model. And for the endogenous biomarker, it was a full PBPK model. And you can also see the clearance type in terms of what we use in the model. Then for the trial design, we use healthy volunteers. The population size was 50. The number of subjects per trial was 50, and the number of trials was 1. So these are all the things that you do in SimSip so that you're able to get a certain output. And then the output will be in terms of the substrate and the inhibitor with interaction and without interaction. So this slide basically shows us the CP1 concentration versus time curve from our simulated data superimposed with our observed data. But this specifically is still work in progress. But this is what we were able to see on day 1, on day 15, and on day 16. So when you put the simulated data from SimSip and you also put the observed data in one graph. But overall, you can be able to note that there was a dose-dependent increase in CP1 concentration with GDCX. This you can see from the colors that you're able to note on this slide. there was no clinically relevant DDI risk, and this is why we say so, because when we look at the dose, the Cmax ratio versus dose graph, we are able to note that below our Cmax ratio was below 1.25 for all the dose levels. You could be able to see for dose level 200, it is less than 1.25. And then when you start going up the dose levels, 400, 800, and 1200, it is above 1.25, but less than 2. So what you need to notice that from guidance from the regulatory authorities, the Cmax ratio, if it's less than 1.25, then have negative DDIs, and there's lack of in vivo ATP1 inhibition, so there's no risk for DDIs. And then if it is less than 2, then you have a weak, you can say weak risk or a small risk of drug-drug interactions. But when you go above 2, then you have a high risk of drug-drug interactions. So from plotting rosuvastatin GDCX, this is what we were able to see. Again, there was an increase in rosuvastatin concentration with GDCX, and this is quite similar from what we see in the observed data again, that there's an increase in CP1 concentration with GDCX. The same is seen with the simulated data here, that there's an increase in rosuvastatin concentration with GDCX. Similar to this graph now combined, that you're able to see it more clearly with this graph. There's a dose-dependent increase in rosuvastatin with GDCX. When you look at the area under the curve, still same thing with what we talked about, the Cmax ratio, is that below 1.25, there's no risk of DDIs. Above 1.25, there's a small chance of DDIs, but this is a weak drug-drug interaction risk. And then when you go above 2, there's a high risk of drug-drug interactions. So from this project, this is what we were able to conclude, that CP1 should be assessed in early clinical development for potential OATP1B inhibitors to confirm or refute DDI risk. And when you use estimated in vivo Ki from CP1 analysis, the magnitude of DDIs for OATP1B inhibitors can be predicted. And so we are just adding a voice to that, whatever is out there. Then dose-dependent increase in CP1 concentration in the presence of GDCX was actually observed, as shown in the graphs. And the estimated in vitro inhibition constant was 5 times the in vivo inhibition constant. And this is what it was in line with what has been seen in literature. So it was within range. There's an increased concentration of rosuvastatin in the presence of OATP1 inhibitor. We could be able to see that, but it was less than 2-fold at the highest dose still. So suggesting that there's limited clinical impact in terms of DDI risk. So these were some of our references. I would like to acknowledge our mentors, Kenta, Yoshida, Sravandi, and from APT, Amira. We also want to acknowledge our APT fellows for always being there for us. I also want to acknowledge the company Genentech. We want to acknowledge Pharmacometrics Africa for bringing us here today, and Satara for together with Satara. So thank you. We're here for any questions.
Thank you, Susan and Sadik. We first start with questions online. Have any questions online? I'll check the chats. So any questions in the room?
To identify a correlation between the impact on CP1 and the impact on rosuvastatin. You see like a 1.5 increase, 1.5-fold decrease in CP1. What would that translate to as far as the impact on rosuvastatin? I assume it's not one-to-one ratio. Or is it?
It's supposed to be similar increase. The trend is supposed to be similar in terms of DDI risk. So if CP1 increases, if you give the drug the inhibitor together with rosuvastatin, you expect to see a similar trend, and you can be able to see it when we begin the presentation.
So it is 1.5-fold increase in CP1. Do you expect a 1.5-fold increase in
In
rosuvastatin?
rosuvastatin. I think I need to confirm that.
I think it shouldn't be exactly the same, but it should be like similar. For example, for this graph here, it shows, for example, for previous drugs, like the probe drug and CP1, it was shown to be similar increase in the CP1 concentration and probe drug. And in our case as well, we have seen the increment in CP1, and we have seen the rosuvastatin, but we haven't really calculated how much they are different, but they should be of similar.
But what I have seen in the compounds I worked on is that increase in CP1 is lower than the increase in the output. I think there might be literature examples for the function. That's good. Because if it's the same, if it is 1.5-fold, 1.5-fold, then you do not need to estimate DDI. Right? You can just say whatever you saw in CP1 is what you get. It's better. And you don't need to go through the exercise, right?
Yeah, that's right. The thing is for CP1, we did Cmax ratio, and for rosuvastatin, we did AUC, because those are the parameters that really captures the exposure. But maybe we do Cmax for rosuvastatin and then the other way around and see how they correlate to each other.
I see usually the most notable changes in Cmax as far as CP1.
Yes. So CP1 is a better predictor for the endogenous biomarker than for the substrate, which is rosuvastatin.
Nice question. Thank you.
Gita, would you like to comment on the question?
Yeah, thanks for the question, and thank you so much, Susan and Sadik, for the very nice presentation. I'm not sure I was able to fully capture the kind of conversation. The voice was not very clear to me, but I guess maybe one point I wanted to make about kind of why do we bother to do the modeling to extrapolate if there are always a correlation between the observation, the CP1 and the probe drugs. That's true when we probably have very cleanly behaved molecule, and also we always follow the same dosing amount or the dosing schedule that we tested already in the clinical scenario. I think when it becomes more important is when we are trying to do the kind of extrapolation to the other dosing level that was not evaluated in the previous studies, or also particularly, for example, in this study, in our first in human study, we have very small number of subjects per dose level. So just looking at the data from one dose level is not clean enough for us to fully characterize the magnitude. So using the modeling is a way to integrate all the information that we have from different dose levels.
Thank you again, Gita, for attending today, and thank you, Saravachi, for your support.